Welcome to the deep dive. Today, we're getting into something really important.
Yeah.
Novel ways to speed up finding biomarkers, imaging techniques, and treatments for Parkinson's, Lewy body dementia, and MSA.
Yeah, and the source material we're digging into is pretty special. It's from a recent conference session, like straight from the experts.
Exactly. So you're getting insights from academics, people from big pharma like Roche, Merck, Eli Lilly.
Plus innovative biotechs, Novus Bio was there, worldwide clinical trials too. They handle the logistics of these big studies.
And a clinician scientist, someone who's really bridging that gap between the lab bench and the patient.
It's a great mix of perspectives for you, covering all the angles.
Right. So our mission here is to really pull out the key takeaways. Where are we now with these diseases? And maybe more importantly, what's next? And one thing that jumps out straight away is biomarkers. They seem foundational.
Absolutely. And there's this interesting historical note the moderator brought up. Apparently, they moderated a similar session 10 years back in Vienna. And Vienna, that's where levodopa was first actually dosed for Parkinson's patients.
Wow. So that really frames it, doesn't it? Progress, but also a long road.
Exactly. It highlights the journey.
Okay, so let's dive in. One of the first big questions was about alpha-synuclein imaging. Why the 20-year wait to see multiple studies finally happening? Rob from Merck touched on this.
Yeah, and what he said was, maybe surprisingly, the biggest roadblock wasn't just the science, the chemistry or whatever.
What was it then?
It was getting access, consistent access to really well-characterized patient samples. You need that high-quality tissue to develop and test these imaging agents.
Ah, okay. Makes sense.
And he mentioned their work with the Michael J. Fox Foundation was absolutely essential for getting that kind of validated Parkinson's tissue. That let them push their radioligand work forward.
Radioligand.
Right.
That's the stuff that lights up the alpha-synuclein on the scan, right?
Precisely. The molecule that binds to it so the PET scanner can see it.
But despite that long wait, things are looking up now.
Yeah, there's a definite sense of optimism. People were talking about recent data from multiple companies and academic labs showing real success.
There'll be a potential turning point.
Could be. The feeling is that being able to actually see alpha-synuclein, track it with biomarkers, might just revolutionize how we tackle these diseases. But it really underlines how dependent this progress is on getting those good samples. Took a while.
So connect the dots for us. This historical lack of good synuclein biomarkers, T tracers, CSF, or blood tests, how did that impact drug development for Parkinson's?
Well, it basically forced the field to rely heavily on clinical endpoints, tracking symptoms over time, how people were functioning.
Right, because you couldn't directly measure the biological target easily.
Exactly. It's well, you used the engine analogy earlier, trying to fix it without looking inside.
And Jose from Eli Lilly made a comparison with Alzheimer's, didn't he?
He did. He talked about this explosion of Alzheimer's biomarkers in the last decade.
Right.
And how that just massively accelerated everything, understanding the disease, the pace of research. The hope is Parkinson's is kind of on the cusp of something similar now.
Which really shows how vital these objective biological markers are.
For the whole field, yeah.
So how is this actually changing clinical trials now? Maria from Novus Bio had some experience here, right, from Alzheimer's.
Yeah, she shared a really interesting lesson. They apparently regretted not using biomarker pre-selection in an earlier Alzheimer's trial.
Pre-selection, meaning choosing patients based on whether they had the biomarker.
Exactly. So for their current Alzheimer's trial, they are using markers, things like p-tau217, brain-derived tau, to make sure they enroll the right people.
Learning from experience, then.
Seems like it. And she was also excited about just the sheer number of new biomarkers becoming available. Even if they select patients based on one marker, they're measuring a whole bunch of others in plasma.
Why do that?
Well, the idea is after the trial, you might be able to see if certain subgroups responded better, maybe because of some other biological factor. It's about getting a more nuanced picture.
Okay. Then Amos, the clinician scientist, he threw a curveball, didn't he? Challenging the whole idea of these being single diseases.
Yeah, that was really thought-provoking. He argued maybe we shouldn't think of Alzheimer's or Parkinson's as one disease, but more like pneumonia. A condition or a syndrome that can have different underlying causes.
Like different bacteria causing pneumonia.
Exactly. He said, we talk about idiopathic Parkinson's cause unknown, that's most people. And even for the genetic forms, like LRRK2 or GBA, we don't fully get how the gene leads to the disease.
So we need to understand the specific trigger in each person.
That was his point. To get beyond just symptom management, you need to know the root cause, like treating the right bug for pneumonia. It argues for a much more personalized approach, doesn't it?
It really does. Okay, shifting slightly. Tom from Worldwide Clinical Trials brought in the practical side. What do CROs need?
Clear definitions. For recruiting patients into trials, you absolutely need robust, consistent diagnostic criteria.
Makes sense for running the study smoothly.
Right. And he mentioned this growing idea that Parkinson's might actually be like 8 or 10 different subgroups under one umbrella.
Wow, really?
Yeah. So that just screams for better, earlier, more precise diagnostics to sort these out. A bit like biomarkers helped refine things in Alzheimer's trials.
Then Fabrizio, the neurologist, made a distinction. Clinical versus biological biomarkers.
Yeah, a helpful one. Clinical biomarkers are the symptoms, things you measure with scales or devices. Biological ones are, stuff in your blood, CSF, brain scans.
Okay. And how reliable are the current clinical ones, like motor symptoms, for tracking progression?
Reasonably reliable for tracking how things change over time in trials, he said. But the big need is for biological markers for early diagnosis, telling Parkinson's apart from look-alike conditions, and tracking the disease itself maybe even before major symptoms show up.
Which leads nicely into rethinking trial design. If we knew 5 years ago what we know now about biomarkers, how would trials look different?
Well, Maria from Novus Bio gave a concrete example for their upcoming Parkinson's Phase 3.
Okay.
They will be selecting patients based on alpha-synuclein using probably a seeding assay or maybe a skin test.
So direct biological targeting.
Right, and they'll measure a whole panel of other fluid markers too. And interestingly, even Alzheimer's markers if the patient has cognitive issues.
So definitely applying lessons learned.
Seems so. And then Amos mentioned this disconnect we often see.
Between lab findings and what doctors see in patients.
Exactly. Like basic science identifies amyloid plaques, but clinically you might see high blood pressure or sleep problems. How do those link up?
Yeah, that gap.
Or how does a clinical sign relate to, say, the size of the hippocampus changing? We often don't know the direct connection. It's a big area to bridge.
Fabrizio then brought up an interesting contrast between Alzheimer's and Parkinson's trial approaches.
Yeah, Alzheimer's trials are really moving towards very early stages, even mild cognitive impairment, right?
Right.
But in Parkinson's, he noted, there's some data, like from the BAB trial apparently, suggesting some drugs might actually work better in more advanced patients.
Huh. That's counterintuitive.
It is a bit. It raises questions about who the best candidates are for certain therapies, like antibodies targeting alpha-synuclein. Maybe it's people who are early, but definitely symptomatic.
And what about the genetic cases before symptoms? That's tricky.
Super tricky. When do you intervene? And how do you even score someone who's prodromal, showing early biological signs? Maybe, but no motor symptoms yet. Big questions.
Van Ken, one of the moderators, floated a more radical idea, biology, as the gold standard.
Yeah, kind of controversial maybe, suggesting that the biological measures should trump clinical symptoms when we assess disease and treatment effects.
So focus purely on the biology, even before someone feels sick.
That's the implication. It opens the door to thinking seriously about trials in these prodromal groups.
Is that happening?
Well, Jose from Eli Lilly mentioned that while Alzheimer's started with early symptomatic folks, they are now doing preclinical trials, intervening before cognitive decline.
And could Parkinson's follow?
He thought maybe. Perhaps focusing on people with REM sleep behavior disorder, RBD, who also show synuclein signs. But the big if is, will treatments actually do anything measurable that early? We don't know yet.
Maria from Novus Bio saw the potential too, right? With CSF or skin assays identifying people early.
Yes, but she raised the practical points. These prodromal studies could be incredibly long, years and years.
And expensive.
Very expensive, especially tough for smaller companies. She also mentioned Roche's approach in their trials, this time-to-event analysis.
What's that?
It's a statistical way to handle the ups and downs and symptoms people get from their dopamine meds, trying to get a clearer signal of the underlying disease change.
Interesting. Okay, back to alpha-synuclein itself. Amos raised a fundamental question. Is it the cause or just a marker?
Right. Like, is it the fire or just the smoke? He used the LRRK2 example. Some carriers get Parkinson's with synuclein buildup. Others with the same gene don't.
Why? It's complex, like the Alzheimer's situation where removing amyloid doesn't always stop the dementia.
Exactly. It feeds back into that idea. Fabrizio mentioned these are probably syndromes, multiple paths leading to a similar clinical picture, so hitting just one target, like synuclein, might not work for everyone.
Though Fabrizio did suggest synuclein might be different from amyloid. more active.
Yeah, he kind of described amyloid as maybe dead material once it's deposited, whereas alpha-synuclein is actively clumping and moving around in cells. So maybe targeting it could have a more direct effect on how neurons are working or not working.
Then the conversation got even more nuanced. Is alpha-synuclein the same in MSA versus Parkinson's?
Yeah, the idea of different strains or shapes, like maybe how it misfolds or gets modified after it's made post-translational modifications, is different in MSA.
Which would mean a drug for MSA might not work for PD.
Potentially, if the target looks different. But Ken pointed out that therapies targeting the mRNA, the instructions for making synuclein.
They might bypass those differences.
Could do, yeah, because the basic instruction is the same. And he also mentioned there's an antibody that seems to recognize synuclein in skin samples from DLB, PD, and MSA patients, so there are definitely shared features too.
Okay, so beyond synuclein, what other targets are people excited about?
Lots of focus on the genetically validated ones, LRRK2, GBA, TMM175, where we have stronger evidence they're involved.
Right.
And also pathways, things like lysosomal function, the cell's recycling system, and mitochondria, the energy factories, plus neuroinflammation.
Inflammation, like NLRP3 inhibitors.
Exactly, and targeting things like interleukins, cytokines. Jose from Lilly also mentioned GLP-1 agonists, the diabetes drugs.
Weren't the results mixed there?
They have been, yeah. But he said earlier studies had some positive signals. The key thing, he stressed, is getting pathway biomarkers.
To match the right drug to the right patient biology.
Precisely. Match the patient, the pathway, the drug, and crucially, the right time or stage of disease.
Which Tania from Roche really emphasized to the importance of disease stage.
Absolutely. You need biomarkers to know where someone is on that timeline, early, advanced, prodromal, to pick the right trial participants.
And are there good early markers emerging?
She mentioned the seeding assays look promising for very early PD and noted that actually diagnosing early PD in trials is often more accurate than early Alzheimer's.
Interesting.
But she also stressed needing pharmacodynamic markers. Does the drug hit the target? And pathway markers, plus better MRI than just the standard B8 scans.
Ken then asked specifically about MSA and DLB. Why test drugs in MSA first sometimes?
Jose explained it's partly because MSA moves faster, it's more aggressive, high unmet needs, so you might see an effect or lack of effect quicker.
A faster proof of concept.
Kind of. And if it works in MSA, that builds a case for trying it in the much larger PD population. Plus, the biomarker situation in MSA is maybe a bit further along in some ways.
Like what?
Higher NFL levels, clearer changes on MRI, maybe MSA-specific PE tracers coming along.
And for DLB, Fabrizio mentioned the pathology overlap.
Yeah, the big thing there is often having both alpha-synuclein and amyloid buildup. That complicates things for treatment development, for sure.
A major challenge seems to be tracking progression in Parkinson's. Is there a go-to biomarker for that yet?
Sadly, no. Not like NFL or GFAP and some other conditions. There isn't one standout progression marker for PD trials right now.
Someone mentioned caffeine levels.
Yeah, an anecdote about one paper, but it doesn't seem to have panned out or been widely confirmed. It just highlights the search is ongoing. And everyone acknowledges the current rating scales can be insensitive, a bit noisy.
So that led to talk about digital biomarkers.
Yeah.
Phones, wearables.
Right. Tania from Roche shared their experience using a phone app in the Pasadena trial, and apparently it showed some promising signals.
How could these help?
Well, the hope is they give you more frequent data, maybe more objective measures of movement, tremor, gait collected in the real world, not just clinic visits.
Could make trials more efficient.
Potentially. Better data fidelity might mean you need fewer participants or get clearer results, but there's also work just refining the traditional clinical endpoints, too.
What about cognition? Especially in DLB and PDD. Are the assessment tools good enough?
That's a challenge. The discussion pointed out that trials often still rely on the standard cognitive tests used for older drug approvals, like for rivastigmine.
But maybe not ideal for DLB specifically.
Possibly not. There's a hope that secondary endpoints could be developed that are more sensitive to the specific cognitive profile you see in DLB.
Fabrizio also touched on how little we understand about how treatments affect non-motor symptoms.
Right.
And the mix of pathologies.
Yeah, it's complex. He made an interesting clinical observation, though, that people with PD who respond well to levodopa early on often seem to have slower progression overall, less cognitive decline later.
Any idea why?
Not really clear, but it suggests some link between that initial dopamine response system and the longer-term disease course. But predicting cognitive outcomes from early trials is still really hard.
Okay. Any other potential biomarkers discussed? What about MRI?
Yes. Tania mentioned exciting results from the PADOVA trial. They saw drug effects on specific MRI measures in the substantia nigra neuromelanin contrast and iron-sensitive contrast.
What does that mean?
It suggests the drug is having a biological effect in a key brain region affected by Parkinson's. So these MRI techniques could become really useful biomarkers for showing target engagement.
Promising, but needs more data to link it to clinical benefit, I guess.
Exactly. Need to see if those MRI changes correlate with people actually feeling or functioning better.
So as they wrapped up, what was the overall feeling? Excitement?
Concern.
A real mix, actually.
Yeah.
Lots of optimism about biomarkers, definitely inspired by the progress in Alzheimer's. People talked about this dawn of the biomarker era moving towards precision medicine.
And new types of drugs. Yeah, excitement about things like oligonucleotides that can target mRNA very specifically. Hope for combination therapies, for finding those distinct PD subgroups for tailored treatments, and just the basic importance of slowing the disease down in PD, since we do have decent symptomatic treatments.
But there were worries, too.
Definitely. Concern about whether we're investing enough in collecting those crucial, well-curated patient samples long-term, that's vital for future biomarker work.
And skepticism about some targets.
A bit, yeah. Some pessimism about LRRK2 maybe not being the magic bullet for everyone, given past trial results. It points back to the complexity. These are probably multifactorial diseases.
Like the co-pathology issue in DLB with synuclein and amyloid.
Exactly. That's a huge challenge. And underlining it all is the fact that we still don't fully understand the exact sequence of events, the precise pathogenesis that leads to these conditions. Still big unknowns.
So wrapping this all up for you listening,
Mm-hmm.
it's clear the field is at this really exciting but also incredibly complex point in tackling Parkinson's, OBD, and MSA.
Yeah, the biomarkers are coming online. Our understanding of the biology is getting deeper. Learning from Alzheimer's is helping. There's real promise there.
We definitely hit some aha moments, I think. Just seeing how intricate biomarker development is, the big shift to precision medicine thinking, how our grasp of the diseases themselves keeps evolving.
And hopefully for you trying to stay informed,
Yeah.
this deep dive gave you a solid overview of where things stand and where they might be heading, directly from the people doing the work.
Which leaves us with a final thought, maybe something a bit provocative for you to mull over.
Given how complex these syndromes seem, with likely multiple causes tangled together, how does research need to change? Do we need to move beyond just targeting one molecule at a time to really make a breakthrough?
And tied to that, what role will massive long-term studies collecting really detailed data from lots of patients over time play in finally cracking the code of these incredibly intricate brain diseases?